First, let's have some background information before we interview Garret Suen. Professor Garret Suen's laboratory studies the microbiome of herbivores. He is specifically interested in understanding how these microbial communities convert plant biomass into nutrients accessible by the host. His group hopes to understand the evolution and ecology of herbivore-associated microbial communities and how they evolve across host types, diets, and geographical areas. Currently, this general interest has led to two specific areas of study: biofuels and animal health and production. First, let's talk about biofuels. Professor Suen's work digs down into the specifics of how microorganisms efficiently break down cellulose and is of great interest for producing ethanol from cellulose. Current biofuel production of ethanol derives its sugar from corn. Using corn is costly, diverting valuable farmland from food production into biofuels. A better solution would be to grow fast-growing plants on low-quality farmland, grind them up, and degrade the cellulose waste to produce ethanol. Garret's lab is working out the details of how these organisms break apart cellulose in hopes of using the knowledge gained for biofuel production. Okay, the second area of study is animal health and production. Ruminants are a major part of the food production landscape, producing milk and beef. Milk production depends upon a healthy ruminant community, as the characteristics of the short-chain fatty acids they make available to the host directly impact the quality of the milk produced. A recent paper by Professor Suen looks at sampling of the mouth, which is buccal swabs, as a proxy for determining the rumen microbiota. Garret realized that since cows chew their cud, which means they regurgitate the rumen contents into their mouth and chew it, the microbiota of the rumen might match the microbiota of the mouth. This paper compares the mouth microbiota to that of the rumen and determines their similarity. What's exciting about the research? Understanding the communities that make up the rumen can help improve cow health and milk production. However, sampling the rumen is difficult. How would you like to go up to a 1,000-pound animal and try to get a sample of its stomach contents? Sampling the mouth is far easier and is a good predictor of the rumen microbiota. It will make possible to measure the gut microbiota of entire herds over time. So, in the paper, Garret showed that indeed they are similar. So a great figure from this paper is figure 1. Figure 1 from the paper shows the sampling just before morning feeding, time 1, results in a buccal swab community, the blue diamonds, that most closely matches the ruminant community. If on this figure, if there's a close match, the symbols will overlap. This supports the idea that buccal swabs can provide a window into the microbiome of the rumen. With some additional processing to remove bacterial strains that are known residents of the oral and feed microbiome, the method becomes even more accurate. So what's really cool about this? A great example of how this information can be used is in predicting the onset of subacute ruminal acidosis. Kind of like GERD, acid reflux in humans. And this is a common problem in milk-producing cows that causes chronic health problems, impairs the efficient use of feed, and increases the environmental impact of milk production. Garret's going to talk about that in this interview and talk about how buccal swabs can be used as a method of early detection of subacute ruminal acidosis, thus treating the cow early and saving suffering of the cow and money. So why should you care? If you like cheese or drink milk, it's pretty clear why Professor Suen's work is impactful. In addition, making dairy farming more efficient and decreasing its environmental impact puts more money in the farmers' pockets, is easier on the land, and creates a better environment for all of us. So let's go to the interview. I am here with Dr. Garret Suen I am here with Professor Garret Suen talking about his work and what he does at the University of Wisconsin-Madison. So I'm going to ask you a series of questions, and then from there you can answer them. Why did you become a scientist?
So I kind of got into science primarily because of my parents. Parents are both Asian, and they wanted me to become a pharmacist, which is a very Asian thing to do. But and I started off. I'm originally from Canada, and I did my undergraduate work at the University of Calgary. And I started off in the biological sciences and realized very quickly that being a pharmacist is not for me. It's not that exciting for me personally. And I actually ended up going into games programming of all things. I did a second degree in computer science because I really wanted to become a game designer. While I was doing that, I was actually working in an AI lab and artificial intelligence, and I fell in love with the research side of things. And that's when I continued on pursuing a master's in computer science. And in that area, I actually studied ants, social insects. They are really interesting, and they form the basis of a lot of what computer science studies in artificial intelligence. And so the idea is, if we can understand so-called lower levels of intelligence and how these creatures are able to basically do many amazing things like build a nest, use their bodies as bridges to overcome obstacles, and there is no one single person telling them what to do. And this principle is a principle of emergence, of how a complex property can arise from the interactions of smaller individuals. That's really what got me charged up and excited about science. I was doing it from a purely computer science perspective, so a lot of modeling on the computers. Then I decided I wanted to actually do the real thing and study ants. So I ended up coming down to the US and doing a PhD at Syracuse University. I'll be completely honest with you, I applied to Syracuse University back in the early 2000s, primarily because they won a national championship in basketball.
All right.
And it had nothing at all to do with really anything. I heard this crazy statistic on the radio that they got $14 million for winning the national championship. So I thought they had a lot of money. Little did I know that that money just goes to the athletic department and not to the rest of the school. Fine. But they offered me a scholarship, so I ended up going to Syracuse University, and I I got interested in this particular bacterium that acts like an ant colony. And this particular organism hunts like ants hunt in real life. And they have these really incredible properties. And so I really got fixated on understanding the microbial side of things. I finished that up, and then I decided that I really wanted to work on ants because I had yet to see a real live ant that you would study in the field outside of your garden variety ant, so to speak. Mm-hmm. So I ended up finding another Canadian, Cameron Curry, here at the UW-Madison, who studies ants, and he studies leafcutter ants. And so I contacted him, and I ended up joining his lab as a postdoctoral fellow. And I eventually got a chance to go down to the tropics in Panama and Costa Rica, got to see leafcutter ants in the wild for the very first time, got to see army ants, which is what I studied for my master's thesis. And we ended up doing a lot of really cool work in that particular arena, trying to understand how the microbes that live in and on the ants help them do what they do. And then I ended up getting a faculty position here in the same department, studying cows, and that's what I do now. So that's really what drove my interest in science. Yeah.
That's a great story. I love it. What is the broad area of research that you work in? To cows, but expand on that.
Sure, yeah. So dairy cows, of course, I think, as everybody knows, is one of the major lifebloods of drivers of the economy here in the state of Wisconsin. And so my area of research is really focused on trying to increase milk production efficiency. So we've probably heard that, cows produce a lot of milk, right? You might be surprised to learn that they're not very efficient at producing milk. Basically, you got to feed them a lot of food and they produce a lot of milk. But the milk actually is mostly water. And what we care about within milk is actually milk protein, milk fats, milk sugars. And that's a lot driven by the demand of the consumer. So these days, when you go to the grocery store and you buy yogurt, everybody is all fixated about Greek yogurt, right? Because it's got a lot of protein. Right? So that's what consumers are demanding. They're demanding products that are high in protein. We produce a lot of cheese. Cheese absolutely depends on milk fat and the types of milk fats that are inside of cheeses. Sugars are another big one. So obviously cow's milk is a replacement for mother's milk in humans. And so sugars are a huge part of that too. And the efficiency at which cows actually produce milk is not very good. And so we're really looking to try and understand that process and try and optimize that. Cows rely on a microbial community that's found inside of their stomach. You probably have heard of the fact that cows have 4 stomachs. The largest of that 4 stomach is the digestive organ, kind of like us and humans. And they actually have a complicated community of microbes that help them digest the food that they consume. So we're trying to understand, are there ways that we can improve digestion so that the cow gets enough, or more, I should say, not just enough, more carbon out of their food, and that they're turning that carbon towards milk production efficiency rather than things like body fat, right? Mm-hmm. You might have also heard of the issue of methane associated with cows and greenhouse gases. That's another aspect too, because in fact, there are specialized microbes inside of the rumen that will take carbon dioxide and convert it into methane. That's actually a net energy loss for the system. And so by improving the efficiency of basically taking that carbon dioxide and turning that towards energy for the cow, you actually remove the production of methane. And overall, that's very great from the perspective of greenhouse gas productions on dairy farms. Yeah, so that's what we're focused on.
Why is your research worth doing?
Well, I think obviously since we're here in the state of Wisconsin and it's a major driver, $43 billion worth, that's a lot of money. And I think if we are able to improve the efficiency of these animals, what that translates into is, number one, a cost in feed savings, a cost in savings for feed for the producer. If we could improve the efficiency, that actually means less cows on a dairy farm, right? So oftentimes these cows are producing so much milk, but a lot of it's water, okay? And so, consumers, the producers, the processors, I should say, the folks that are converting the milk into cheeses and yogurts, they don't want to pay for water, right? So if we can improve the efficiency of these animals to put more fat, more sugar, more protein into their milk, right? That means less water. And so you're not just wasting, the feed that's being produced. That could translate into less animals on dairy farms. That's a lot easier for farmers to manage because these days, unfortunately, farms in Wisconsin, the small family farms are disappearing, and you're starting to see large giant co-ops, such as what you see in California. These are 3,000 animals. Managing that number of animals is just really difficult. And so if we can reduce the number of animals that these farmers are managing, that saves a lot of energy, a lot of management, in addition to land usage, right? So if we can remove the large footprint of these farms, that is part of the reason why we think that this research is worth doing, in addition to other aspects such as mitigation of methane. If we can reduce methane that these cows are producing, that should, in theory, reduce the overall amount of greenhouse gases that are floating around in the environment.
Discuss a recent finding that you're really excited about.
Sure. So as a microbiologist, one of the things that a lot of people wonder is, okay, so this whole new term of microbiome, right? Everybody's talking about the microbiome, but what does it really mean to apply microbiome research in real life, right? So, in the medical field, we've talked a little bit about this. People are asking those questions, saying, "Oh yeah, things like fecal matter transplants, right? Where they're an actual therapeutic." Well, what about an agricultural aspect? And that's really what we've been trying to push. So one of the big ones that we've been trying to really develop is this concept that your microbiome is a reflection of your overall health. This is very much true in a dairy cow. So some of the major diseases of dairy cows are driven by what's happening inside of your rumen, inside of their stomachs. Great example of this is a condition called subacute rumen acidosis. What that happens is, kind of like acid reflux, that rumen becomes very, very acidic. When that rumen becomes very acidic, what happens is the animals get very sick. It is often leads to milk fat depression, which means that the animals are not capable of actually putting fat into their milk. It's very uncomfortable for the animals as well. Anyone who has ever suffered from acid reflux knows exactly what that feels like. So these are what these animals are dealing with. Research that has been done has shown that the microbial community inside of the rumen actually begins to shift very early on when that condition is starting to develop. Currently, there are no really good ways of diagnosing the onset of subacute rumen acidosis. And the way that people do it now is they wait for the symptoms and then they treat the symptoms, right? So you can't prevent it. You can basically only treat it when you actually get it. And one of the big things in the field would be if we were able to, in fact, diagnose this condition even before it develops. The antidote for this is really simple. You just simply shift the diet, because it's actually a dietary effect. So during different parts of the life cycle of the cow, you switch diets, right, depending on their metabolic needs. Sometimes that shift in diet can cause this particular symptom. And so if we can detect that even before it happens, then it's a simple shift for the veterinarian to say, okay, I think we can just change this diet and stave off the development of that particular condition. And we think that the ability to do that is really a strong application of the microbiome. And so what we've been pioneering is this principle, or this idea, called buccal swabs, which essentially you've probably heard of the term of the cow chewing the cud, right? Well, what chewing the cud is the cow is regurgitating material from their rumen, and they are processing even further. So in humans, the way we bite is like this, but cows don't do this. They do this because they are grinding the grains that they consume into smaller and smaller and smaller particle sizes. And so they regurgitate the plant material that they've consumed, they grind it into smaller and smaller particles, and they swallow it again. And so what we've been doing is pioneering this approach where we basically take a swab, and it's no different than like swabbing the inside of your cheek, but you do it to a cow. You just swab the inside of the cheek of the cow, you take that back to lab, and what we can do is we can actually figure out the community of the rumen from that simple sample. Okay? And this is, we think, quite groundbreaking, because fecal material in a cow looks nothing like what's inside of the rumen. So unlike humans, where your fecal material actually is a good representation of your microbiome in your stomach, in a cow it's completely different. And so one of the biggest challenges is, how do you actually sample that rumen community? And this is one way that we think is very effective. And so what we've been doing is running trials and tagging on with other people who have been doing trials where they are looking at subacute rumen acidosis and sampling these animals, a week, 2 weeks before the actual onset of the condition, and seeing if we can track the changes in that microbial community, and when is that inflection point where you see the community diverge so much that, okay, that's the time where we could actually come in and make some type of an intervention to overcome an issue with these animals. And so this is a thing that we're trying to pilot and get going, and we're hopeful that that will not only be useful for subacute rumen acidosis, but many other metabolic conditions that these animals endure, negative energy balance, ketosis, and things like that. So,
that's really neat. Thanks for sharing that.
Yeah.
All right. What class do you teach, and what is the subject?
So the class I teach is called Micro 657. It's a graduate level course, and the focus of the course is, it's called Bioinformatics for Microbiologists. And so what is bioinformatics? Well, so bioinformatics is the incorporation of computer science into biology. So I mentioned earlier that I've got dual degrees in computer science and biology. And as we are now in the age of genome sequencing, where we can sequence any piece of DNA, it now becomes a big challenge to actually manage all of that data, right? And so there are a lot of techniques that have been developed specifically to, analyze DNA. And so that's the course that I teach. It's essentially, you can think about it as basic computer science for biologists. How do you navigate on a computer to be able to manage these large data sets? How do you construct databases? How do you use those databases to find the information that you're looking for? And that's the course that I teach.
Yeah, and the information you're looking for is like genes.
Correct.
And
yeah. Genes and proteins, right? And what are their potential activities? Yeah.
Okay. And then finally, what are your interests and hobbies outside of work?
So probably my biggest passion outside of what I do here is travel. I like to say to a lot of people that I actually got into this gig that I'm doing now for the travel, because as faculty, we get to travel a lot to different conferences. So Yeah, I am a huge art buff, and so I have it on my bucket list of I have a lot of bucket list art things that I do, like seeing every single Vermeer in their home art galleries, or one of the biggest ones that I'm slowly crossing off. I'm very close. Is Van Gogh has he painted a whole set of 12 different sunflowers, the famous Van Gogh sunflowers. He has 12 of them. 11 are in existence, and you can go to the different galleries to see them. And so I think I'm about 2 away from seeing all of them in person. And so that's the kind of stuff that I like to do. I like to create little bucket lists of things that I do, and I incorporate that as part of my travel. And so that's what I do. That's what I love to do for fun. Is we are constantly traveling all the time. We have an 8-year-old, and we drag him with us wherever we go. So his passport is quite full of stamps and.
Oh, fun! That's fun. A lot of great memories.
Yeah. So.
All right. Thank you, Garret.
Yeah,
I really appreciate it.
Sure.